Okay, so let's start and thank our audience for joining for another talk in this by CCPBioSim series of industry talks. Today we are delighted to have Richard Gowers as our speaker, and he's going to talk about a topic which has become very important for industry, which is MD simulation and their analysis. So this title talk is MDAnalysis and Alchemical simulation, which is definitely relevant to drug discovery and industrial work. Richard is a software engineer with a history of working on projects related to drug discovery, and he has previously worked at NextMove Software and the Open Free Energy project. And he has been contributing to the MDAnalysis project for over a decade. And I think that he's going to explain to us what the MDAnalysis project is. And so the floor is yours, Richard. Thanks again. Many thanks for accepting to give this presentation.
Cool. Thank you. Thanks for the nice introduction. I'm guessing the screen share is working?
It is, yes.
Cool. All right, I'll crack on then. So a quick outline of what I'm going to try and cover today, if I have enough time. I'll give a quick introduction to the MDAnalysis project as a whole. So if you've not heard of it before, don't worry, because I'll hopefully give a good introduction. I'll highlight a recent initiative that the project has been funded to do, which is this MDA kits idea, which hopefully might be the next generation for sort of molecular simulation analysis. I'll then give some examples and highlights of how I've been using MDAnalysis recently for my own work, which was the work I did at the Open Free Energy project on alchemical free energy calculations. And finally, if I've got enough time and I don't run over, I'll get into a bit more of kind of future developments for MDAnalysis and things that we are working on that you might see soon or that might be coming up soon. So just to quickly introduce the MDAnalysis organization, the MDAnalysis is a Python package which is built by scientists for scientists. It's a community-led development of this package. It's not really based in any one institution, but is instead a distributed group of developers from various institutions across the world who contribute to this Python package that's now sort of branching out into several sub-packages, as we'll see. The majority of the development of this project has actually not been funded. So I've been working on it for about a decade now, I think 11 years. And it's only been in the last 2 or 3 years that we've actually got any money for the development of this package through the Chan Zuckerberg initiative. And we've been doing sort of various mentoring schemes as well. So we've been doing Google Summer of Code and outreach along to that. So, I mean, who is MDAnalysis? To date, there's been about 205 different code contributors to the package. This is, I think, a chronological list of everybody that's ever contributed. A few names in there are bolded if they've sort of done various or some significant contributions. And there's a few of the funders at the bottom that have been involved with us over time as well. In a nutshell, this is sort of what MDAnalysis tries to do. So on the left, you have some sort of simulation trajectory that you've produced with your favorite molecular dynamics program. This often gives you files which are in a variety of different formats. There's a brief list of them there. So you could have sort of BCD, XDC, TRR files, various files, both for the trajectory and also the topology information for your system. And this causes a bit of a headache because you have sort of all these different file formats, and it's just a wild west of different file formats. But through the MDAnalysis Python package, you can sort of coerce all of these different file formats into a single platform, a single framework. that then lets you access all of your data primarily in NumPy arrays, which is sort of is sort of the lingua franca for dealing with numerical data in Python. And then you can manipulate these NumPy arrays through various analysis algorithms to sort of produce processed data and images and graphs and all sorts, which hopefully then leads you to sort of insights and publications on the right there. And sort of along the bottom here, you can see sort of from greater than sort of 40 different possible file formats, you can hopefully use the single analysis script on any sort of input to give you your processed data, which is nice, and it's sort of leading into ideas of sort of interoperability and being able to share your analysis work with different researchers. I'm going to try, I have tried to not put too much code into this talk, but just as a quick example, if this is sort of what using MDAnalysis would look like, so you can kind of import it and then you can put in your different molecular simulation files. So here we've got like a PDB and XTC file from GROMACS, and then you can access the positions of your atoms there, and you get given out a NumPy array. So hopefully it's as simple as that. So the project's been running for, I think, since 2011 or so. I think it started either at Oxford or Johns Hopkins, I forget, I wasn't there. So it's been open source since January 2008. We've been sponsored by NumFocus since January 2020, where NumFocus is a organization which is hosting various scientific packages, scientific software packages, including sort of NumPy and SciPy and others. Most people that start using the project eventually sort of lean into starting to develop it sometimes, and those developers have then formed the leadership of the project over the years. I think we try to claim that we're the most used Python library for molecular dynamics analysis nowadays, based on sort of citations. My own involvement started, I think, in 2013 when I was doing my PhD, and I was sort of writing my own MD code. And at some point, the MD code started working. So then I had a new problem, which is I had to analyze my calculations. And so I sort of probably typed something like that into Google, and it brought me to this Google Code website, which Google Code was what you used before GitHub was really a thing. And so I came across this Google Code site, and I got in touch with Oliver there, who was running the project at the time and still is. And I started to sort of contribute code there. So that's sort of my own involvement has been since then. So I'll sort of jump into this initiative that we've got going recently, which is this NDA kits idea. So this has been funded by the Chan Zuckerberg Foundation, which has this EOSS project, which is a quite good source of funding for open source science. And so we got this funding here, and something we've tried to do with it is build out this idea of NDA kits, which is a framework for fair, compliant molecular simulation analysis, which is trying to sort of enable people to package up their own analysis code into small toolboxes, which are building upon MDAnalysis and sort of achieve a specific aim. So a sort of a scaling problem that we've encountered as a project is that with sort of greater than 200 code contributors and community members, most people often write code in MDAnalysis and they sort of want to contribute this back to the project, which is obviously a nice problem to have. But over time, this has become a bit of a scaling problem. So there are advantages to this in that sort of the developers of MDAnalysis can help to, ensure this code is of high quality if it becomes centralized and sort of it becomes available to everybody once it gets sort of accumulated into this core. And it can kind of continue to be maintained over time if this does happen. But this centralized solution has sort of grown a bit unsustainable over time because I think the developers at the core of this all became a bit overloaded with trying to sort of maintain all of the code at the same time. And there's sort of ideas about sort of bloats with code bringing different dependencies, It can be a bit of a barrier to people contributing if you have this sort of minimum quality the code has to hit. And similarly, a lot of molecular simulation analysis code can be quite specialized, and so it's quite hard to maintain all of these different quite specialized tools at the same time. So this is sort of the problem we're trying to solve with this MDA kits idea, where we sort of created a framework where people can create their own packages. So this is sort of the core developers in the middle here that have developed this framework that people can people can create their own packages. So we created a cookie cutter template, which is sort of a template where people can create their own Python package based upon this template. And they can then sort of publish their own software within that package. I think there's a review process that we can sort of help people with and sort of will assist people in making these mini packages. We've got sort of an internal registry or an external registry, sorry, of all these different packages so people can kind of look up and find different analyses tool, which are sort of part of this kit registry. So this is sort of then making it easily findable and accessible. The structure of this cookie cutter package that we put together hopefully encourages people to follow best practices when they're creating these packages. So hopefully people will be producing software that is usable by other people if they're sort of following the guidelines that we've set out. And we're hoping this is a solution to this scaling problem that we've hit of trying to maintain a centralized repo that has everyone's code in all at once. So I'm going to sort of show a few examples of how this is taking off and sort of where people are using this and what this has led to. The first example is we have this MDA Hold 2 package, which is for the analysis of pathways through molecular channels. The problem that we're facing with this one is this whole package is quite an old piece of code, and so getting it to compile is always a bit of a challenge. But you don't want to sort of have this in your core repository. So we sort of put this out into a MDA kit where people can now download this in a compiled form through the Conda Forge infrastructure. So another example, which is something we actually did through a with a student that was doing a mentoring scheme with us, is this ProLif package, which is the protein ligand interaction fingerprint, is what that stands for. And this is good at generating these diagrams that you can see on the right here, which are identifying sort of pharmacophore interactions. This is something that Cedric has produced. And this has, again, been packaged up into an MDA kit that people can use and download. And then finally, as a more recent example, we've had this solvation analysis code come through by Orion, who's over in Berkeley, if I remember right. And this is work that he's been doing, I think, on material science and looking at ions and batteries and things like that, which has then led to this being published in the Journal of Open Source Software as well, which is quite nice. So this is a nice example of some code that grad students produced that's sort of gone through to be published and sort of get the recognition that it needs. So this MDA kits idea is I think it's becoming quite successful. It's a nice way that people can package up their code into a way that other people can find it and use it. And it's hopefully going to start to become an ecosystem of these small packages that you might see coming up when you search for how to do your analysis. Okay, so I'm going to move on to talk about some of my more recent work on analyzing FEP calculations. So a problem I was facing a while back was I was working on this OpenFE project, which was building an open source pipeline for the prediction of binding free energies. Okay. And the final section of this workflow was to do some analysis, primarily to try and detect sort of bad calculations. Often you can detect when a calculation has gone wrong by looking at the structural information there and sort of how the structure of both the protein and the ligand have changed over time. So because you're running quite short simulations, if you see that your protein conformation has changed quite a lot over time, that probably indicates that it wasn't stable to start with, or the thing that you've sampled isn't quite correct. And similarly, if you see that your ligand has moved quite a lot. in that time and potentially drifted out of the binding pocket, then you probably haven't sampled the right thing, and it might be a bad simulation. And sort of a key criteria for this was that everything had to be automated, because you were looking at running hundreds, if not thousands, of these calculations. And so you needed your structural analysis to give you a way of immediately flagging bad results in a sort of a reliable and a robust way. So the problem with this, the problems I was facing with this particular data was the input data, which is generally in a PDB-like form, that is often quite chaotic, the files that you're given in that format. The data produced was from sort of a molecular simulation program that was outputting the data in a bit of a strange format. And similarly, analyzing the ligand motion, as we'll get to see, was you had to sort of think hard about how you were analyzing if the ligand was moving, and sort of what do you consider movement of the ligand. I think I've got a quick video here of what a alchemical free energy calculation does. So here we've got sort of a small ligand in a binding pocket, and we're interested in seeing this sort of fluoro group growing on the side there. And so that's sort of the process we'll be looking at. You can see if the with or without that fluoro group would be more would bind stronger or not, would be the basis of the calculation you're running. So firstly, looking at sort of input sanitation, a problem that you often have is you're given these sort of PDB files or PDBX files, and they are often, everything's often mislabeled. So there is quite a strong standard for how these files are meant to be used, but that standard is often sort of followed to varying degrees, and people have their own sort of variants on that standards. In particular, the names that are given to sort of atoms and residues are often sort of vary between programs. You might find that different programs are giving you different canonical names for different residues. This isn't really a problem that's been solved with the new PDBX format, which is shown here in the bottom, which is sort of meant to be the future of this file format. Another problem is these files often don't give you all of the bonded information for your atoms. This is sort of meant to be inferred or taken from sort of external data sources, which if things in your files aren't labeled correctly, it's a bit of a problem to infer which information you're meant to be pulling into these files. So we had to essentially beat this data into shape before we could start to analyze these calculations. So the protein data bank, the PDB, does provide a database of possible components which you are expected to encounter. It's in this resource called the Chemical Component Dictionary, which you can download in those links. I think this is about 300 megabytes when you download it, and there's sort of a few 100,000 different components that you could encounter, from sort of standard amino acids to post-translational modifications to sort of various other small components that you could find. And so on a good day, you can then just sort of quickly do a cross-reference from things that you find in your input files based upon the residue names to sort of components that you find in this chemical component dictionary. But on a bad day, that's sort of not so easy because, like I said, things are often mislabeled. And so a solution that we had to build out was how do you search this chemical component dictionary with sort of a fuzzy input, because you know the thing that you're searching for, you know that the thing you're starting with, the sort of the data in your input file is slightly wrong. So then how do you sort of cross-reference this against this chemical component dictionary? And so what we found was often the input data was correctly labeled in terms of the elements that were present, and you could often guess the bonding information based upon sort of the van der Waals radius to sort of guess which particles are bonded to each other. And from that, you can get an element graph. You can then build an index of this chemical component dictionary, which has the element graph smiles for each component. So here on the right, I've got an example of a tyrosine and how you can turn that into an element graph, and then you can sort of produce the canonical smiles for that element graph. And that's then just a string, which you can kind of look up in a fairly fast time, no matter how large this database is. And so you can then sort of iterate through your input file residue by residue, you can generate this element graph representation of the residue, and you can try and cross-reference that against the index that you've built of the chemical component dictionary. And then once you've found the correct template, you can then pull back the sort of more detailed information, such as sort of the bond orders and the stereochemistry and the more particular, or the stereochemistry labels and the more particular atom names. So we've packaged this up into a small package called PDBinf, which tries to sort of be a bit more smart at loading PDB files and doing this table join between the chemical component dictionary, which is sort of the canonical source of truth, and sort of these quite messy PDB files. And so hopefully when you're loading things like a phosphothreonine there on the right, you'll be getting sort of the correct bond orders and not just sort of this incorrect sort of form on the left. So these correctly labeled files, it seems like a bit of a sort of distinction to say, like, do you have correct bond orders and are the atom names correct? But it's actually quite important for various steps in molecular simulation, such as when you're trying to apply force fields, having the correct bond orders is quite important for the OpenFF, the open force field sort of variant of force fields, which are doing smarts matching to apply force field parameters. Or if you're applying sort of the AMBER family of force fields, you need the correct atom names to get the correct parameters applied, for example. So this sort of package helps with both of those. And similarly with post-simulation analysis, it's quite handy to have your correct atom names, because then it's easy to find your psi and your chi dihedrals, because you can select these based upon atom names. This is currently based upon RD-KIC. It's a bit easier to do some of the smiles matching and smarts matching that I was talking about before, but we'll be bringing this into MDAnalysis soon, probably in the form of an MDA kit, actually. So if you're working a lot with PDB formatted files and you're getting a bit frustrated that it's not correctly formatted, or you wish you could sort of do some more complex analysis, then this is something to look out for. So a second problem that we encountered was everyone's favorite, which is periodic boundary conditions. So something we'd often find was when we sort of went to visualize our simulations, like we've done on the right there, it would look like the ligand was sort of off drifting in free space, whereas the protein was sort of in the corner of the simulation box. But this is just an artifact of how the system has been centered. So the protein has been put in the origin of the simulation box there at sort of 0, 0, 0. And this means that it's quite easy for the ligand to actually wrap into a different molecular image. So while these two things visually appear quite separate, they're actually, next to each other once you consider periodic images. But this makes it quite difficult to do some types of analysis, and sort of it makes it just a bit of a headache to work with. But you can quite easily fix this using MDAnalysis via the on-the-fly transformations module, which lets you essentially pre-process your trajectory as it's read it. And so you can kind of re-image molecules onto each other as you're sort of streaming the trajectory in, which is quite nice. A more kind of complex example of that we then hit was the protein as a whole might be sort of slowly translating or sort of rotating or tumbling in the simulation, and this would give the illusion of quite a lot of motion when you're sort of considering the motion of the ligand inside the binding pocket of that protein. And you kind of want to ignore that sort of extraneous motion of the protein when you're thinking about, has my ligand drifted, because you kind of want to only consider motion relative to the binding pocket. So a quite neat solution we found to this is that if you can subtract the motion of the protein from the simulation, you then sort of create a new reference frame that you're sort of analyzing everything within. So you can kind of subtract the center of motion mass quite easily from frame to frame, so sort of the center of mass of the protein remains fixed over time. And then we found that we were able to use this QCP method, which I've got cited down below, to calculate the rotational matrix that would it's the rotational matrix that would minimize the RMSD over time. And so if you just apply the opposite of that rotational matrix, or yeah, if you apply that rotational matrix, you can also nullify any rotation that the protein has done from frame to frame. And so sort of by negating those two forms of motion, you can actually keep the protein fixed over time, or and you're applying these transformations to the entire system, despite the fact they're calculated on the protein. And this then gives the illusion, or the sort of the effect, that the protein is fixed over time on average, and then sort of any motion that you're then seeing in the ligand is hopefully meaningful after those transformations. So as a quick example of how this works, it's a fun example to try and explain this. When they originally created the Star Trek series, I don't think they had spaceships that they could film in. And so what they would have to do is they would have to sort of do camera tricks to try and make it look like the spaceship was sort of flying and being hit and things. And so the actors would then have to sort of dance around the set to sort of give the illusion of motion. And so a fun trick you can now play is if you kind of fix a certain frame of reference within a sort of a camera shot for Star Trek, you can then sort of see that it's not actually the ship moving, but it's actually the actors sort of jumping around on the set. So here they've kept the part of the ship fixed, and you can see the actors sort of jump around. And this is sort of the same trick that we've done with the protein, where we've sort of kept the protein constant, and hopefully then you can see the motion of the ligand over time. And so we kind of did this all in MDAnalysis using this transformations pipeline, which is sort of a filtering pipeline. And there's various steps to this where you can unwrap each molecule to make sure that bonds aren't being broken over periodic boundaries. You can then image different components of the system to make sure everything's in the same image in periodic boundaries. You remove any center of mass motion, and then you can remove any rotation of the protein to hopefully give sort of a stable reference frame. So hopefully I've sort of shown how MDAnalysis can be used in sort of a more industrial setting, and a more sort of a fully automated workflow. If you're interested in this, it's all open source, and so you should be able to sort of look it up at this OpenFE_analysis package, which has all of this. If you're interested in applying this sort of transformations yourself, you can kind of look up how those were done. And I think kind of this application of this sort of reference frame stabilization made the rest of the analysis on sort of looking, is the ligand moving over time, and sort of what's the RMSD of the ligand and things like that. It made it quite simple to do by doing a bit of sort of pre-processing on the data. Okay. And so then finally, I'm going to sort of move on to a bit more of a pet project of mine that started off as a bit of a lockdown project that's sort of taking up a bit too much of my time recently, called Dystopia, a lockdown project gone too far. So as I've sort of said, MDAnalysis is a Python package, which is a nice language to work with, but for certain time-critical parts of the package, so sort of where you're spending a lot of compute time, we then sort of have to compile certain parts of the code. So we sort of write things in C++ and sort of bind to that via Cython. On one example of this is when you're doing kind of distance calculations between potentially, hundreds and thousands of different atoms. And these distance calculations are the kind of the workhorse of various other analyses, such as RMSDs, radial distribution functions, mean square displacements. And so it's not very glamorous work, but it's also the engine that keeps a lot of these other things running. And so this is work that my friend Hugo and I have been sort of working on and off since the pandemic. And so something we noticed was that the performance of these distance calculations routines wasn't as fast as it could be. And we started to sort of dig into why this could be and how we could improve the performance of this. It was a bit of a optimization pass. And so to understand the problem, there's sort of two key pieces of kind of techie, computer sciencey things that we have to sort of get our heads around. The first of these is this idea of sort of vector instructions, which is if I have a loop which is adding together many different positions, a modern computer processor can actually add together multiple of these items at the same time. in what's called a vectorized instruction. This is a little bit different to parallelism. This isn't using multiple cores, but this is just a single core can hold, let's say, 4 different numbers side by side, and it can add 4 different numbers to 4 different other numbers, all in one instruction. And so if you do that correctly, then it should go 4 times faster because you've done sort of 4 additions with 1 instruction. So this is what's called vector instructions. And then the second sort of key piece of information is that when you write code in C++, it's the job of the compiler to then sort of look at your code and make improvements to your code, and sort of hopefully improve your code and produce these vectorized instructions. And so the output of a compiler, so when you sort of run GCC, is going to produce sort of this assembly language, or will produce a object code which you can understand via assembly language, which mostly corresponds to what a CPU kind of actually executes. So it takes your C++ code and turns it into a different sort of code, which is what the CPU actually executes. So at some point, Hugo and I sort of looked at this compiled code, and we saw that these nice vectorized instructions that hopefully make things run a lot faster because they're doing kind of multiple things at the same time, weren't being created by the compiler. And so this was sort of where we started to focus our attentions. I promise this is the only slide with assembly in it, but so when I talk about sort of inspecting the code that's produced, you can do this on this website called Godbolt. So here I've got sort of a very simple distance array code on the left here, which is going to calculate a matrix of distances, and you can kind of see what a compiler would produce here on the right-hand side, and it produces this sort of different representation of this. And so after a while, you can kind of inspect this and see kind of what instructions are being produced. And in particular, we're seeing here that the vector instructions that we're hoping for aren't being produced. This is kind of quite nicely color-coded as well, this output. And so if you spend like an afternoon looking at this, you can kind of figure out what's going on with the different instructions. But if you're curious, you can look into this. This is why I have the slides. And this is sort of what we did. And we found that the correct instructions weren't being produced. And so, what we ended up then doing was, if the compiler wasn't going to produce the right code path through us and wasn't going to produce these nice vectorized instructions, you can then sort of write the assembly code directly, or you can write the SIMD intrinsics, as they're called, directly to sort of bypass the compiler. So there'll be sort of a block of code which the compiler doesn't optimize, but instead just sort of passes through kind of object code that you've directly written. Is a bit of a simplification of what's happened. And more specifically, we've done this using the Google Highway library. So if you're interested in looking into this yourself, I would recommend that library. It's actually producing kind of multiple code paths at different vector widths. And so as sort of a way of testing a hypothesis that this should be improving things, is that we compiled the code on a sort of a single machine, and we ran the same code with increasing vector widths. So we sort of first ran it 1 number at a time, doing the addition sort of 1 number at a time, then you can kind of add 2 numbers at the same time. And you should see that your performance doubles as you keep sort of increasing the vector width from sort of single scalar values to sort of slightly wider vectors, to AVX2 vectors, to AVX512 vectors. And this is the graph that we have on the right-hand side here. And we are seeing that sort of performance is sort of jumping up as you increase the vector widths, which is showing us that the producing the code which has these vectorized instructions is providing extra performance. There's not as much of a jump up for the AVX512 instruction set as we'd like, and we're sort of looking into that. But overall, it's looking quite promising. So this was we were quite happy when we saw this. And so the overall thing here is that as part of this lockdown project, I think we've created the fastest distance calculation backend for molecular simulation. As sort of a bit of a fun project. We've kind of compared it against a few of the molecular analysis codes there on the right-hand side. We've gone from being that sort of orange line on the top, which was previously the slowest way to do this, to that blue line on the bottom, which is where we're hoping we can kind of get the performance of this. I think we're currently at the purple line because there's some overheads that we can't quite get rid of yet. So we're quite happy with how this has turned out, and we're sort of finally using the computer hardware effectively. This is gradually being incorporated into MDAnalysis, but it's also a standalone library because all it's working on is just C arrays. There's nothing inherently MDAnalysis about the data we're considering. So it's actually a standalone package. So again, if you're looking at doing this sort of analysis and you want a fast backend for calculating distances with minimum image convention, then you might be able to check out this package. Cool. And so that's everything I had